Very good. Thank you very much, Jason, and thank you, everyone. Good morning, good afternoon, and good evening. We've got a pretty decent attendance. It's an interesting topic. The challenges faced by many of us in the conduct of clinical trials are many, and we're all striving towards improved quality of data and efficiencies and how to get our studies done quicker for our mutual customers, the sponsors. In this scenario, what we would like to do is share a real case study that we participated in. We've desensitized this for obvious reasons, but we wanted to share the learnings with the broader community as we feel that some aspects of what we encountered and how we solved the various problems may be applicable across the board. The background of this case study is one of an organization that had pretty solid CRO core imaging experience, and they were looking to expand that and offer a broader base of solutions as a core lab. The organization had, because of its history, had a number of really well-refined proprietary systems, and this is not uncommon for many of the core labs that we know, that because of the history of technology, many core labs took it upon themselves to develop their own image management system, in some cases their own analytic tool, and so forth. However, from the inception of those technologies, the overall industry, e-clinical industry, has come a long way. So this one particular scenario, they had these investments and they wanted to maintain them and continue them. They were very happy with those platforms they had developed in-house. They were tailor-made for their use. But at the same time, because they wanted to expand their services, they also reached out and they did an evaluation of a number of off-the-shelf tools. They were looking for tools for project management and clinical trial management, and obviously for image analytics. Philosophically, they followed a very sound practice in the sense that they knew what they were very good at, which was the conduct of the trial. They were very good at getting radiologists, and they were very good at the clinical aspect of things and data management and project management. However, they really didn't want to also become a high-end IT technology shop. So they chose a CTMS off-the-shelf and an image analytics tool built by Mint Lesion. And I'm very happy that Matthias Seitel is here with us to describe to you how Mint Lesion and the image analytics tool fit in and what its capabilities are. And the combined was to form the platform for them to become the foundation. Without the technology tools, they were facing a number of challenges. And some of these challenges are pretty common across many of our operations. It doesn't mean that we do poor work. It just basically means that we have to spend twice as much time perhaps looking at the data. We QC the data many times over, and as a result we spend a lot of labor doing so. So it's not that everything is in total disarray, it's just that it's not highly optimized. So in this particular case, CRFs were being manually collected. In many cases, they were reports that were coming out of one system or another, getting loaded into an Excel spreadsheet and then sent around as email attachments. This is something that, again, we have seen is pretty common in the industry. And when the data had to be consolidated and combined, oftentimes it would take too long for everyone to get the data together. Sometimes emails had gone missing, sometimes paper was missing, sometimes the spreadsheets were inconsistent, and that was giving rise to a lot of labor being expended, double checking things and combining and scrubbing the data, as I like to call it. And finally, the ultimate customer that we all strive to satisfy, the sponsor, we could not be responsive to them. You know, very often they're very anxious, more anxious than we are, to see the results of our study and to see where we are and how much progress we've made. Are the sites recruiting fast enough? Are the readers reading the time points fast enough? Well, providing that input to sponsors is pretty much probably a big part of the responsibility of project managers in most of our organizations. And they were always told that everything was going to take some time, that if from the time they would put a request in, for whatever reason, it would take a couple of weeks, and then the data had to be collected and scrubbed and so on. And then internal project status reporting was also a very laborious task. In other words, to have an effective management dashboard in-house, we all need to know exactly where everybody stands in terms of the reads and the sites that we have and where in the process all of the reads are. So even for internal management, these reports were manually constructed. And finally, in many cases, as data handoffs from one system to the next was happening, sometimes in paper form, sometimes in CDISC formats or comma delimited format. It doesn't matter what the format is. The minute you're talking about handing off data from system A to system B, even if you don't modify the data in any way, we all know that there are chances of transcription errors simply because there are hands touching the data. And that was one of the things that you will see we eliminated very effectively. No hands was touching the data from the point of entry all the way through to the sponsor, and that made a big difference. So in short, each individual tool was doing its job well. They were missing some elements, namely they were missing a robust analytic tool, image analytic tool, and they chose Mint Lesion for that. They needed something to track some of the financial aspects, so they decided to buy a CTMS system and interface it with their financial system. But altogether, all of these pieces were still costly to maintain because there needed to be labor and time layered on top of these things to have the data move across. And naturally, as we all know, that's not very efficient and it is error prone. The challenge that we were given by this client was basically, as I mentioned, eliminate all the hand touching of the data. And moreover, we took things one step further, and I will talk about that during the course of the demo. Moving data from point A to point B is something that we've been doing for a long, long time. As I described earlier, it was stilted and it required hand-holding, but it is something that we have been doing. What we were able to do was make that real time. We were able to make it bidirectional, and we were able to conform to the industry standards that were required for that. So the total package that had to interoperate seamlessly consisted of two commercially available components, Mint Lesion that you'll hear about a little later, the CTMS tool. There was a proprietary image system, which was theirs, and there was a EDC, open source EDC, which is actually a variant of what we offer on our platform. And then there was our interoperability platform, which I'll describe a little later. It's the hosted platform as a service model. And all of these things were cloud-based, by the way, and they all had to be C-disk compatible, because clearly they didn't want to introduce proprietary new data formats. So that was a very important thing for us that we accomplished for them. The way this was done, if you can see my mouse, at the bottom right-hand corner is our adaptive eClinical bus, which is a cloud-based interoperability platform. And I want to make a distinction between interoperability and integration. As I mentioned, we have all been doing integration for a long time. The extract, transform, and load approach has been around for a long, long time. What is novel here is the real-time transactions moving from down below here where the image storage is and the analytics tool and the readers and investigators sit. The data traveled in real time, and it would splinter off, and it would update the CTMS so the project managers could see exactly what progress was being made. They would do their scheduling of reads through here. And then multiple CRFs were being created simply because the way you view data after a read or during a read sometimes is not necessarily the way you want to view the data when you're doing data management. And in the demo, what I'll be showing you is a pretty complex workflow of a dual read adjudicated study. And the elements here were the PACS system was the proprietary image store. The analytics tool was Mint Lesion. The CTMS was from Bioptronics. And as I mentioned, the data management was the EDC. The way that was done was through use of our plug-and-play connectors. We have a number of these connectors that we have already pre-certified to work with the most popular tools out there. So let's say for EDC, they had told us they wanted to use Medidata. We would have simply dropped out the connector for this EDC and dropped in the one for Medidata. And we have connectors for the most popular eClinical vendors such as Bioclinica, Medidata, and so on. Same with the CTMS tool for Bioptronics. Now, material for a future webinar, we also have connectors to medical devices, and we also have connectors to analytic tools. This is data analytics such as Spotfire and Tableau. And then the latest salvo of connectors that we have developed have been for electronic medical records, which is something that is in great demand these days. And as many of you know, FDA has shown a keen interest in eSource and EMRs being an eSource. And of course, ePROS, IWR, and IVR is also something that we work to integrate. However, this particular case study and the demo were concentrating on these three legs: the investigator read, project management, and the EDC. And the final point here is this is a fully validated platform. Buying technology, as you all know, sometimes is a lot cheaper than making sure that it is in compliance and it remains in compliance. So that was another big part of the project, was not only how to assemble all of these components and get them to interoperate, but it also was how do we maintain the CFR Part 11 e-signatures? How do we have the audit trails that show the source and destination, who modified it during the course, from what system? And all of these things had to obviously be validated and traced and so on and so forth, which was all done in record time. Um, because of the uniqueness of our technology and our platform and our accomplishments, we just recently were recognized by Pharmatech magazine as one of the top 10 e-clinical trial management solutions, and we were featured there. We're very proud of that. So what were some of the gains? So we put all of these effort in with a great deal of participation from our clients and the other vendors. The first thing is, in a model where you're a core lab and you're conducting the same type of studies time and time again, once the interoperability is put in place, it is highly reusable. And at least that's how our platform operates. So we were able to accomplish some 70% reuse of the design components when it came to the EDC. As we all know, as a core lab or a CRO, if you get two studies that appear to be identical, invariably there are differences. So I'm not sure if we could ever get to 100% reuse, but realistically I believe that we can get better than 70%, but 70% was a major coup because prior to that everything was scratch built. A long time ago I was at a conference and the analogy was made for interoperability in clinical trials as wanting to place a telephone call from New York City to San Francisco, and every time you want to place the call, you lay down the wire, you set up the telephone poles, and then once the call is over, you take down the wire, you take down the telephone poles, of course the analogy of the telephone call and the clinical study, and then for the next one you rebuild everything. So clearly that's highly inefficient, and we made that go away for them. And because of all of these reuses of these modules, study setup time was reduced significantly. Again, what is the strength of all of us service providers is the clinical capabilities that we bring to the table. So inevitably protocols are always different. However, by and large, we were able to achieve some reduction of 68% of the study setup time. Data management, just not having to chase down paper or spreadsheets and being able to immediately see from a dashboard. They were able to save a lot of their time and be able to dedicate their time to more important activities. And of course, source data verification. Again, this is eSource is a topic for another one of our webinars that's upcoming. There's a great deal of focus on that. But it's intuitive that when you eliminate intermediate copies of the data, that you will, not need, you will save a lot of time and not need source data verification. And finally, reduction in resources. This was fairly modest, simply because the staff that were there were really working a lot of extra hours, and they were able to have a little bit more of a customary sane schedule. So we had seamless data interchange amongst all the systems and components by the study. The whole thing, by the way, from beginning to end, took us maybe a couple of months. So this was not an 18-month effort, simply because our platform is already built and it's already set in place. The couple of months was really spent in interviewing data management staff, clinical staff for the protocol, just to make sure that the data transformations that we built into the rules engine were done right, and then the subsequent validation and user acceptance. But really there was very little coding involved here. In subsequent studies, they specifically RECIST, they were able to knock down the study setup time to less than 2 weeks. They eliminated data errors due to re-entry. And this next bullet, real-time on-demand status reporting, that really was utopia for them as well as the sponsor. Just being able to see a up-to-date snapshot of exactly where everyone is at the site level without having to wait for data management or project management to handle the data. Of course, as a core lab, you have complete control of the visibility of the data, but just knowing that you have that option to you yourself look at the data in real time, and in turn filtering the data and presenting it to your sponsor was a great thing for them. One of the things in the rules engine that I mentioned was we also codified some of the clinical activity. So this was some of the time point determinations, some of the study-related determinations, as well as some of the workflow aspects that was dictated. And because of that, there was consistency in how all of that was done. So during the course of the audit that we participated in along with our client, the auditors were very comfortable understanding that here is a system that does something consistently, and it was based on a set of requirements that was visible to them, and it no longer had the variability of all of the different people perhaps interpreting the data ever so slightly different, having to trace their training and all of that. And really the technology aspect of the audit was completely successful. The balance of the audit also was very successful. And the final point is that you know, sponsors like us understand that with interoperability you are more efficient. They understand that with interoperability you're more cost effective, and they understand that with interoperability the data that you produce is a lot more accurate. And as a result of these automations, they were able to win a number of new studies just simply by presenting how effective everything interoperated. What I'd like to do at this point is hand this over to my colleague Matthias Seitel from MINT Lesion, and he's going to describe a little bit of a background about the company and then go into a demo of the product. Thank you.
So hello everybody, and thank you very much, Sina, for inviting us as MINT Medical to be part of this webinar here and have a brief demo of our product MINT Lesion. Before going into an actual product demonstration, I would like to cover a couple of slides to explain a little bit about the background of our company and of our main product MINT Lesion. MINT Medical was founded in 2009 as a spin-off of the German Cancer Research Center in Heidelberg. We are currently approximately 25 scientists and engineers working directly at MINT. MINT Medical is owned partly by the German Cancer Research Center itself, but also by scientists coming from this research center. We do offer certified medical product software, and our main product, as already said, is MINT Lesion, a platform for medical image analysis with a strong focus on oncological cases and especially also clinical trial medical imaging in oncology. Our MINT Lesion product, since its beginning, is a CE-cleared medical device. Since 2011 and renewed also in 2015, we have a FDA 510(k) clearance, and we also adhere to various international standards like the Part 11, CGCP, CGMP, and also certain German and European ISO and other norms. To tell you a little bit about the background and the capabilities of our software, as you can see here in the screenshot, MINT Lesion offers similar functionalities in terms of image analysis and display as a conventional PACS or imaging platform would. We offer analysis of a couple of different imaging modalities, of course, such as CT and MRI, diffusion MRI, but also modalities such as PET, PET-CT, SPECT, or also photographic images. And each of these modalities is supported via a specific protocol and measurement and hanging capabilities. A strength of our product definitely lies in the automated response calculation based on various response criteria. RECIST 1.1 is just one of an entire list of criteria that we offer, and I'm going to talk about that a little bit more in a bit. Another strong focus of our product right from the beginning was to focus on time sequence reads, where not only a single time point is being analyzed, but an entire range of time points, as of course is often the case in an oncological read, where you have baseline, subsequent follow-ups, and maybe even another baseline being defined as treatment lines are being changed. A fundamental principle of our software design, and you will also see this in our demo, is to analyze the context of the reader as he is going through whatever task he has to work on. Depending on whether a reader would work on a RECIST case, for example, or on a case with other response criteria such as CHESN, or would even do some clinical work, for example, a tumor staging task, the basic user interface of MintLesion always remains principally the same amongst these different tasks. Nevertheless, we offer a couple of different user guidance approaches and also customizations of the way how the software presents itself in order to assist the reader to perform the task that he is working on, and not to kind of deviate from the task by asking questions that may only partially be relevant. The outcome of this approach is always a report, be it in a visual form such as a PDF as shown here, which can consist of a tabular representation of the data entered into the software, or making use of graphical schemes, or displaying images as they are captured by the reader, or it can also be in an electronic format. We support various interfaces for exporting the data to other systems, what of course is very relevant when we are collaborating with the adaptive system in order to make a smooth transition of data from one system to the other. MintLesion, as I already mentioned, offers a range of different response criteria for clinical trial work. So we offer things like RECIST in both the old and the new standard, IR-RECIST, CHESN, RANO, various variants of the M-RECIST criteria, and so on and so forth. Each of these modules is very flexible in terms of configuration, so there's a great possibility of taking individual parameters of these criteria and modifying them to the actual needs of the trial that you would be working on. In addition to that, we offer a very flexible workflow engine which would allow for a very fine-tuned definition of a workflow within a clinical trial, the definition of, for example, 2 blinded reads or an adjudication. In addition to that, the possibility of defining different user roles, for example, project managers, readers, adjudicators, or sponsor users, all of them having different accessibility levels for the software and also different possibilities of entering data into the application. We do have possibilities for raising data queries, for communicating to readers, to remind them about ongoing tasks or to pose questions simply, and also the capability of defining and capturing adverse events, protocol deviations, and such alike. In the background, every action that is performed in the MintLesion user interface, especially critical actions such as sign off of a time point, but also things like the setup and creation of a trial or different management tasks, are contained or documented in an audit and security trail that can freely be exported again into third-party systems that can be filtered by a certain trial that you may be monitoring currently. And in addition to that, all the data that we capture and that we can export into other systems can also undergo a certain workflow. So for example, when a read is finished, when the reader submits the data and signs it by his signature, by his electronic signature, this would be a trigger for pushing the data to another system or for storing it on a hard disk or in a PAC system or whatever is the need of the respective study. At this point, I would like to switch over to the actual MintLesion application to give you a more visual impression of what the software is about, and later on also how it can interface to the adaptive system. So to start with, I'm logging in as a project manager user. When I'm doing so, I get presented with the first screen of the application, which is giving you an overview over all the subjects currently in the system. The project manager has access to pretty much all the data that is there, so he's not blinded from anything. He has the possibility to manage clinical trials, so we currently have these 2 clinical trials defined within MintLesion. I could add, for the purpose of this demonstration, another trial, and define what reads should come under this particular trial. So I could say I have a read 1 and a read 2, for example, and for both reads I choose the RECIST 1.1 response criteria for automated response calculations. This will then appear as another folder, here displayed on the left side. And now when I select a patient from my list of patients, I do have the possibility of adding this patient to the clinical trial, of course. By doing so, I will automatically create time points for each of the DICOM studies that are currently available for the patients. These are displayed here. I can remove certain of these time points if they are not relevant for the purpose of this discussion here. I can assign readers from a list of defined readers to a certain trial or to a certain read, and this read would then appear in the work list of this reader, and I can clear individual time points for the actual read to be conducted. After I have done so, let's assume my work here is finished and I'm logging in as the reader user that I just assigned this case to. My presentation of the software is in principle quite similar, so I also have the manage screen as we have seen before. However, I have only limited visibility in this default configuration of the user's set of permissions. I only see the patient's ID, and I only see all the patients that I currently have reads to perform on. So I can select my patient and I can select the trial that I need to work on. Now I will only see in the baseline time point. I do not yet see the subsequent time points cleared for reading. Switching over to the read screen, which is the second screen of the application, I have, as I said before, basic image viewing and rendering capabilities. I can choose from the different series or studies that are associated with this time point. I could change the hanging in order to compare different series available at current. And of course I can also perform measurements in order to quantify anything that might occur in this data. So obviously this patient has a couple of metastases in the liver. So let's take a look at some of the tools that we offer here. We do offer automated 3D segmentation tools where I simply draw a small region of interest around the tumor that I want to segment. Then automatically in 3 dimensions the lesion is segmented and the longest diameter and its short axis is derived from that. And this is particularly important because we are dealing with the RECIST criteria. I can pull up a little explanation about the criteria and how they are implemented in the software to get further details. After I have done this, I have the possibility to choose the anatomical location of that particular lesion, so that would be liver in this case. And I can choose whether I want to classify this as a target lesion or as a non-target lesion in accordance with the response criteria. So I put this over to a target lesion category and then I can proceed with performing the actual measurements. For example, maybe taking a 2D measurement tool for capturing this more diffuse but very large lesion here on the top. And basically perform the same process as before, classify this as liver, pull it over to the target lesion category, and that's it for that particular lesion. For brevity, I of course won't work on all the lesions here, but let's take another example of this relative small lesion down here. I can also encircle that and classify this again as liver. And now when I take this and pull it over into the target lesion category, This is possible in the MINT lesion software. However, I get a conformity issue being raised by the software, and this is displayed up here. I can click on this view button here in order to see more details about it. So it's RECIST 1.1 specific. It's a conformity issue against RECIST 1.1. On the one hand, we do have currently more than 2 target lesions defined for the organ liver, which should not be the case according to RECIST. And also the diameter, the longest diameter of the lesion that I just classified is only 9.5 millimeters, and therefore below the 10 millimeter threshold that is defined as a minimum size requirement by RECIST. So now I could go back and simply revisit these lesions and decide what lesion I want to move into the non-target lesion category instead. Of course, this would be the third lesion in this situation here. Now the conformity issue has vanished. Okay, when I'm finished with reading this time point, I can proceed to the third screen, the reporting screen, in order to get a tabular summary of what I have done, which is relatively simplistic in this case. I just had 3 lesions defined. I can add additional comments in this field here. And I also can proceed, if I'm satisfied with everything, with the sign-off of this particular time point. Now I get the sign-off dialog, and it is telling me that the read is actually not entirely complete because I did not finish the image quality check. I was not able to submit the data because I was not working on that image quality assessment so far. So this is a possibility that is offered by MINT lesion. I can choose the image quality, for example suboptimal, choose another rationale for that, and then close it. And now, since I have answered this set of questions, I have the possibility of going back to report screen and being able to actually proceed with the sign-off. This is very configurable, so I have the possibility, for example, of configuring a trial in such a way that sign-off is not possible until all the conformity issues, like we have seen a couple of examples for before, are resolved. So this is configurable on a trial basis. Now let's assume we are complete and my time point read is finished. In the background, now what happens is an electronic PDF report is being generated for that particular time point. I do not have access to this in my capabilities as a reader. However, a project manager or another person able to view this data would be able to pull that up, and we can have a look at this in just a bit. Going back to read, I see that there's now another time point coming up. After I've completed baseline, I have unlocked my access to follow-up 1, and by default this is displayed in a comparison view. On the left side I still have my list of lesions as I initially defined them, but now they are stated here for time point follow-up 1, and because I did not yet measure them, they are classified as not assessed here. So what I need to do, I need to navigate to the corresponding slice, at least approximately. I already am in the correct position here at the patient. The measurement tool that I used, that baseline, already is pre-selected, so I simply can, for that particular lesion, perform the measurement, and it's now registering together with this TO1 liver finding that I already had. Down here you see a timeline where you can see how this particular lesion evolved over the course of baseline and follow-up 1. I can proceed to the other lesions and perform measurements just in the same way, determining maybe in this case the slice with the longest diameter, perform the 2D measurement. This is registered together with the lesion, and this is probably everything that I need to do. For non-target lesions especially, I have the possibility of qualitatively assessing the lesion. So I could, for example, say that this lesion is disappeared. There's still something here, but let's assume for the time being this was disappeared. I can simply choose from this drop down here in order to indicate this, and this again will be considered in the response evaluation algorithm. Also what I can do at follow-up is define new lesions that might have occurred in the meanwhile. While I think this patient does not have a new lesion here, let's just assume this was a new lesion, so I can mark this, for example, with an arrow. Oh, sorry, this was wrong selection here. Have another finding created, classify this as liver, and then pull it over into the new lesion category. Again, going to report screen, I see these measurement values being displayed here, and also in this bottom part of the screen I have the possibility to see what kind of response outcome this is resulting in, which is progressive disease in this case. I have a stable disease for the targets, I have a new lesion present, and this is speaking for progressive disease. Okay, to fasten this up a little bit, and also to switch then to the part where we demonstrate the bridge to the adaptive clinical system, I have, as a preparation, also performed a more complete read consisting of baseline and 3 follow-ups for the same patient. So let's log out here and log in back as the project manager. I select this particular case. So I have now more access to data of that case. When I switch to the report screen, I see the entire table, as I've seen also as the reader. But in addition to that, I have various possibilities of further displaying the data. For example, displaying charts to show how individual lesions, or also how the sum of the lesions develops over time. I have the possibility of exporting this data in various ways. I can also open up PDF documents. This is pulling on my second screen, which is just an example of how this data can be transformed into an electronic document, displaying various graphs, snapshots of the lesions, and also a summary whether there are any violations to the response criteria that I currently use. And of course, I also have the possibility of pushing this data to other systems. And I think this is the point that would be ideal for making the transition back to Sina, who will then show you how the data received from a lesion can further be displayed and post-processed in the adaptive clinical system.
Okay, very good. Thank you, Jason. Thank you, Matthias. So what I have on my screen is the Adaptive eClinical's dashboard, or the EDC. It is a full functioning EDC, and what I'm going to log in as is a data manager. So Matthias did an excellent job of showing you the experience of the individual reader. Now, this interface, or this EDC CRF structure, was set up based on the data management plan that the client had requested for this particular study. And the way you manage data as a data manager for a holistic study is sometimes different than the actual requirements for an individual read. So you will see a lot of the parallels, for example, the dual read adjudication. So we have we created and we have support for individual roles where you can only see the records that are assigned to you. So this is reader 1, reader 2, and then the adjudication and so forth. In the interest of time, I will kind of go through, but just know that this is in every respect a full-blown EDC with all the capabilities. It can stand alone and function well. So I'm going to go to the subject matrix. Each row is a subject, and each column corresponds to an activity. So here we have for the subject called GXB 120, which if you had good vision, you probably saw the patient ID there. So a couple of things happen. When Matthias hit the save key on Mint lesion, the record was created here for this particular reader, and the information was de-identified because on the Mint side, for good reason, they had some information there that was not necessarily in terms of the subject's identification. And then a number of data elements were moved over. The baseline had to be kept separate, and baseline appears in a separate CRF by itself, and I'm going to show this to you real quick. And again, the arrangement of the data when you're doing data management is sometimes different than when you're doing the read. So in this case, the data was appearing in tabular form. So this is the subject information up here. It's a baseline time point. It's RECIST 1-1. This information came from the DICOM header. I'm going to go to the lesion tab. And the results of the lesion identification exercise targets and non-targets in the baseline, the short and long axis data, and then the reference diameters. The target sum of reference diameters was an important data point that we actually dynamically calculated. So this is an example of something that was important for data management and clinical, but really was not something they wanted to clutter up the reader's space. Now, not all of the data elements were relevant, but here is, like, for example, the lesion level comments was important, and finally messages on the lesion level. So this is the what the baseline CRF that was created looks like. And as Matthias showed you, there were 3 unstudied time points, and to keep the view compact, each has a single CRF, but it shows up with this little multiplier that you see here. The status is carried over from Mint, so data entry started as they sign. On Mint side, it comes over as signed as well here. So all of that audit trail that Matthias talked about carries over here, and whatever manipulation to the record takes place here is also audited, so that chain of custody is traced. So I'm looking at the first follow-up here, and I'm going to go to the lesion now. On a unstudied time point, you may have new lesion as well. So this, as you can see, this is a considerably more complex ECRF than the baseline. The target sum was still a required calculation. However, some of these sum of reference diameters carried over, the baseline changes were carried over as well. And then in addition to that, the time point responses that Matthias showed you, they all came over as well. Rearranged because data management felt that this was more compact and more efficient for the way they look at it. They kind of prefer to look at it in a tabular form. Now, one other thing that happened was, and again, in the interest of time, I won't show you the other time points, but just know that 3 CRFs were created, 1 for each time point. Now, what data management also wanted, and this was just for administrative purposes, was they wanted to have what they called a summary view, so they could, for each subject, see a snapshot. So here it is, all of those CRFs and all of those reads condensed into a simple table. So there is the date of the time point, and this is the time point response. This is the best overall response based on analysis of all of these. So this is actually something that Mint does, but in this case we calculated this on the fly. So this is a perfect example of the clinical rules that we mentioned to you. And then the date of first response. And those of us who are radiologists in the audience know that you know, the RECIST determination of the best overall response and confirmed best overall response is a pretty complicated algorithm. And this is what we were able to codify. Saved them a lot of time because they were doing this manually. And then the other thing that they wanted to do was they actually wanted to allow for clinical data review, okay, of the same study read. But in this case, they also wanted us to, again, determine the best overall response based on the clinical data read or review. Okay, so the idea here was the data that comes over from Mint comes over, gets condensed, and then the best overall response. But then when they do it manually, if clinical data review was needed, the same thing, and that's how they were able to cross-tabulate. I'm just going to describe a little bit more about some of the capabilities at this point, since we're coming to the end of the webinar. But for adjudication, there is a workflow for adjudication. So for example, let me quickly show you. If you look here, there are three. There is a reader 1, reader 2, and if I switch roles to the adjudicator. Now, if I had logged in as an adjudicator, I would not be able to switch sites. But here you have the adjudicator's view, and they're not seeing any of the intermittent reads. And the only thing that they see at the end is the adjudication CRF. And here what we had to do was a couple of things. One was we didn't want to say reader 1, reader 2, so we randomly juxtaposed the results from each reader to avoid an adjudication bias. And then the adjudicator would see the results in terms of the best overall response, the confirmed best overall response, which again, if you remember, we calculated, and so on, and data progression, and then they would just check off the agree. So all of this data makes it into the EDC. And now, very quickly, I just want to show you the final part. Now, once you have the data in aggregate form, you can now look at the data and generate reports for your sponsor. And these are reports that are time batched. In other words, they can go out real time or they can go off at night at any point in time. And then you can choose the actual fields that you include. And I'm going to just quickly show you all the different export formats. So from HTML to the screen to tab delimited SPSS CDISC, we have 4 different varieties of CDISC here, and then SAS and so on. So as you can see, we eliminated a huge number of steps involved. All of the normal data management capabilities are also available through this EDC, and this EDC actually is available at no extra charge. We felt that it was very important. In future webinars, we can also show you how this data then moves from this data warehouse to analytic tools such as Spotfire. We can also show you how this data can seamlessly transfer over. Like you can keep a copy here and put a copy into metadata EDC. So that would be a scenario where, for example, your sponsor uses metadata and you don't really want to have all your staff trained and relearn metadata, or sponsor might have the requirement of some exotic EDC that you have not encountered. All you do is you stick with the training that you have had to use this EDC, and you leave all of the data transfer and transformation over there. So what we showed you collectively was all the capabilities of Mint Lesion. We also showed you the capabilities of how data got transferred over to the Adaptives EDC through the bus. We also showed you all of the transformations and the clinical. Time check, I'm getting the hook here, so I want to turn this over to Jason. Jason, how would you like to proceed? Do we have time for any questions, or can we postpone questions later?
Well, we will have to, it's beyond the hour, so we want to be mindful of people's time. Thank you, everyone, for being able to join us on our webinar on how to improve the accuracy and speed of your oncology studies. And we hope that you were able to gain something from the discussion. We're going to leave the contact information up on the screen here if you need to reach out to us personally. Hopefully we can answer any questions that you have. We noticed that there are 1 or 2 questions that have been asked here, and we'll have somebody reach out to you personally regarding that as well. So thank you, Sina. Thank you, Matthias, for both the insight and the demonstration. And thank you, everyone, for being able to join us.